Scott. Urgent question before we start the general discussion debate. For Scott. Yes, please.
Scott, thanks so much for a great talk. Always, it's a pleasure listening to you.
Thanks, Alex.
I did want to ask one thing. You touched on the characterization, but not on efficacy. And how much does that actually enter your considerations when you even come to look at different projects or when you look at a future product?
Yeah, so that's a great question. So relevant to the NCL specifically, you have to have demonstrated efficacy in an in vivo system in order for us to even characterize it. We simply do not have the resources to go after efficacy for each and every submission. It would exhaust, it would drain all of us, because keep in mind also that you have a specific indication that you're going after. So we have maybe 12 animal models, and we'll try to fit into that. We have a metastatic lung model, but again, it's the onus is on the submitter before they come. They make the case to us that excuse me, that the efficacy is fairly solid, and our job is going to do characterization that's relevant to the CMC and the farm tox portion of the IND. Well, that's a lot of acronyms.
Thank you.
Okay.
Okay, then, Scott, thanks again. And I would like to, there's time for discussion, time for debate.
Do we sit down?
And who can I give the floor to ask questions to the speakers? I would like to do it the following way. The speakers, well, there's no place here to sit, and I don't ask you to stand there. So if you're approached, or if your approach or a question is asked to you, then speakers stand up and participate in the debate. I think that should work. So who is there to start first? Well, when you start, you're first anyway, isn't it? Okay, then I'll take the liberty to kick off. And I would like to go to, well, a number of people brought it up, critical quality attributes. That is an interesting concept. What I find more interesting is the follow-up, and only one person showed the follow-up point there, and that was Catherine, and she didn't do it today, but yesterday. Critical quality attributes. They are this particle size, zeta potential, surface characteristics. I don't care. I mean, they are words. There are no numbers. And to really do something with these critical quality attributes, you need numbers. Is 10 millifold, I mean, Scott made the point, 10 millifold different from 12 millifold? Should I disapprove something like that, or should I accept it? And Catherine mentioned the words, the design space. How do I come from a critical quality attribute to a design space? Who is willing to address that point? Okay. I'm sorry, but I think it's a fair question, isn't it? Because there is, that's the point. Is 10 millifold different from 12 millifold? Can I accept it, yes or no? Scott. So please stand up, and you can turn your back to me, that's fine.
Okay. So as I very quickly went over in my talk, the CQA, by definition, influences a biological effect. So if you want to look at a table from a regulatory agency that says your liposome has to be 120 nanometers, well, they won't do that because it's on you to show that I can make a liposome between 110 and 130, and there's no biological effect. I can make that very easily, routinely, so I don't have to do exhaustively of looking where that bottom boundary is at. Is it 30 nanometers? As long as you can reliably make batch-to-batch consistency on your 120 nanometers, you're good. I mean, I can't speak for the FDA, but if you have a very new concept and you've got fluency on all the physical chemical parameters and you know how each and every one, or not each and every one, but the majority of ones influence a biological effect, either efficacy or safety, there's actually quite a bit of room.
I haven't seen in literature anything on NBCD quality on the design space. So challenge me. I challenge you, so tell me that I'm wrong. Where is the data? I know 2 publications where they look at the design space. One is the AMAP, the other one is Trojano, and that's it. And, excuse me?
It's all proprietary.
That's the point. So it's proprietary, but how do I know, what does the FDA, 120 nanometers, 122 nanometers, is acceptable? You have to run a lot of experiments there, and I don't see it in the literature. And I would like to see these data. What is now unacceptable, or what is the design space there? I would like to see it, but I don't see it, and I miss that. Okay, I don't want to, Catherine, you want to say something more than what Scott was saying? Otherwise, I move to other issues, because I think I'm now heating up the audience, isn't it? I mean...
So I just want to follow up to what Scott did mention. I mean, so when you talk about, what is acceptable in the design space and how you choose that, we do see that. I agree that probably a lot of it doesn't go to the literature, but because it is proprietary. But when we talk about discrimination, and we talked about that somewhat in the in vitro release test, making sure that it's discriminatory between things that are in spec and out of spec. Well, how do they do that? For something like a manufacturing process, they can mess up intentionally the manufacturing process and make sure that their tests see that. So that data, I mean, is available. It sounds like it's not available publicly, but that's one of the ways that's approached.
Okay. Yeah, Marissa?
Well, we've been working with the quality by design for a long time, as you know, and of course the data are not public on proprietary figures and numbers, but I think in Europe we have the so-called totality of evidence approach. So let's assume you have a product and where you have a sort of range of variability in the size of your compound, and this variability, this range of size goes along with the batches you use in all development phase, which reflect also the marketing lots in a certain sense. If this variability has not created a truly measurable impact in all in vitro, in vivo, clinical, whatever, certain elements will become the parameters on which you design your space. So the acceptance of ranges rather than fixed figures is pretty common in the biologicals, for example. In the biologicals, it's extraordinary how much flexibility you have in the range, because at the end, what counts for us is the totality of evidence. If a product with certain fingerprint, 5 species, 6 species, is consistently the same 5, 6 species all along, and the results are positive in terms of safety and efficacy, that's fine. This is the space in which you can move. I don't know whether this is too simplified, but I think this is the actual bottom line. The totality of evidence is the only way whereby you can actually accept certain variability in the products themselves. And we have been obliged to do that all the time with the DNA recombinant. We are now doing the same with all the advanced therapies, genome editing, you mentioned cell gene therapy products. There you never have one figure, never. You always have a range, and you have to look at how this range behaves at the various steps of the development. I don't know whether this is the answer you wanted.
Well, first Henning, and then I'll round it up.
I didn't want you to leave this particular point because it's an essential point. And I think we should not generate data, we should generate information. And that means, what is the meaning of certain differences? And the responses were all for generic or similar or follow-up situations. But what happens if the innovator changes the procedure? And I have in mind what happens in the biosimilar field. And there was a situation that the innovator changed the product. And I said, oh my goodness, how can he do that? Because we know the concept is the product is a process. If you change the process, you change the product. And the response from the company, I asked them, was, well, we did not expect such a market success, and therefore we had scale-up procedures. And the generic competitors, they did a very interesting approach. And they reviewed and analyzed, I think, over 7 or 8 years, the batches from the market. And they found differences and said, well, that is obviously our design space. And as long as we are in between, then it's fine. And my question was to the authorities, how could you accept all these changes without asking for new clinical data? And therefore, it's too easy to say, well, we see the entire totality of evidence. If there's a change in the innovator product, you are, Scott, it's not the responsibility of the applicant alone. You have to say, well, do we need new clinical data or not? And therefore, I think this is an essential point.
Yeah, sometimes you might think that Henning and I are sort of have a pas de deux, but that's not true. It's not set up in advance.
Thank you. I think it's a quite very good discussion in that way, and this totality of evidence, which I think is a very good approach. But what I do not understand always is, even at the point when you do a market authorization, the totality of evidence is not yet there, in a way. So you have the pharmacovigilance and so on. And in contrast to new drugs, where if something happens, the withdrawal could be one of the issues to be addressed. But I have not seen that with these complex drugs follow-ons up to now. What is the reason why? Is the vigilance not good enough? Running into clinical trials, again, it's so difficult. If you look into the statistical power, you have tremendous trials to do. And do you find what you want? So again, for me, how to approach that issue, and is there not a need to go a little bit more strict into the period after market authorization? And then we know we have bad data sometimes coming back.
Marissa?
I'm afraid I have to respond.
Second time?
It's obvious I cannot leave this unrecorded. And there are two elements. The first one is that normally, when there is a change in the manufacturing process of an approved drug, we have the so-called follow-on scientific advice. I have no recollection of companies who have had the first registration that they change something and they don't come to discuss very much into the detail what's happening with the change first. Second, the totality of evidence does not necessarily imply a randomized double-blind clinical trial. So it depends on the totality of evidence which is needed. Because I think, particularly with the biologicals products, you know that you can have a range like batch to batch, not even before and after change in manufacturing, batch to batch, quite significant variability. But based on what we have at the time of registration and afterwards, because remember there is always the sample testing, there's a lot of activities we carry on around the, let's say, post-authorization oversight. We have the evidence to say, okay, this has been this change, there's this critical quality parameter which has been altered, but on the basis of the studies which have been submitted, because they will always be submitted, and we can ask them to be submitted, even there is no variation request, then we can confirm that the benefit risk is not altered. So would be, I think, inappropriate just because some pharmaceutical parameters or quality parameters have changed to push towards investing in a clinical trial. Sometimes the clinical trial is not even the best sensitive method. Actually, clinical trials are pretty difficult to pick up little things. So this is why the comparability goes much earlier on than the actual clinical trial design. In some cases is needed, but I think I have to check whether there's been 1 or 2 cases in which additional studies have been required. But as far as I know, not really double-blind randomized accompanying equality variations. No.
We all agree, I think, that clinical trials are not necessarily the best way to prove similarity. I think that I
Yeah.
think we all agree with that. So I don't want that point to be sort of hang around. I'm still waiting for the audience to participate, but the audience sitting there enjoying the debate among these people here, which indeed indicates that there are issues around there. But Mike, you first.
I was just going to comment to Marissa's point that Health Canada also does, basically they're called notifiable changes. So if there's, Health Canada has notifiable changes. So if a company changes its manufacturing process, they have to submit a notification to us that they've
I don't think that was the point Henning was making. The point Henning was making is you notify, and how do you know that it is the same? And in biosimilars, there are very nice, well, in biologics, there are very nice examples where the product really changed. And we don't know whether comparability studies were made and what kind of comparability studies were made and what the outcome was. I think that was Henning's point, but maybe Henning can talk for himself.
As far as I remember, that was adalimumab, and that has been published, and there were significant differences. And my question is, when do the authorities see a signal for a major change, or whatever, how I should call it, to ask for additional evidence that this is clinically non-significant? Because if this is not the case, if you are sure that the original version and the changed, or after variation, the product after variation, are clinically equivalent, then I think the generic approach is fine, that they say, well, as long as we are somewhere in between, then we are on the safe side. And I am not sure whether this is the case, and it's not only a question of scientific advice in advance, it was the outcome of the batches before and after the change or the variation which differed. And I think I was surprised of the magnitude of the difference.
Ladies and gentlemen, it is 10:20, it's 10:21, I understand. We started on time. I would like to close on time because you really need some coffee, which is a very complex product, by the way. But and there are we should run comparability studies because not always the coffee is that good. Here it is, of course, excellent. The title of the session was Global Clinical Experience with Non-Biological Complex Drugs and their follow-on versions. Are we closer to regulatory alignment? There's a question mark there, and I leave it up to you to take out the question mark or to leave it there. I thank all the speakers for their contributions. It's obvious and clear that there's more discussion to come, and definitely next week in Washington we will continue with a number of you the discussion there. Thanks for being here. Thanks for being a good audience. Thanks for participating in the debate and discussion. And that is my last, ironical. And now it's up to my co-chair to.
Thanks, Dan. I will not lengthen with that. Just if you're interested in Budapest meeting, we have here some circulars. Take it home. Give it to other people who might be interested. At least for the people in Europe, it's an easy way to go there. Thanks so much, and have a good coffee break.